Assalamualaikum and hi again, I'm Dr. Nafis Ahmad Kamaruddin from Biology Unit as per UPM, and this is Topic 6 Metabolism, Energy and Enzymes Part 2. Previously in Part 1, I talked about the flow and forms of energy, metabolism and metabolic pathway. So in this lecture or Part 2, I'll explain on the enzymes and the organelles that are called the energy-related organelles. Enzyme is one of the most important molecules of life. As mentioned before, enzymes involved in metabolic reactions. In each step of a metabolic pathway, there is a specific enzyme that catalyzes the reaction. Previously, you learned in school that enzyme speeds up a chemical reaction by lowering down the activation energy. In this lecture, we'll go deeper into the structure of enzyme, how it functions, and how it can be affected by many factors. So first, the structure. Enzyme is a protein molecule formed from the folding of a long polypeptide. Enzyme has a specific site called active site. This small part of the enzyme is where a specific substrate can bind to. There is also another site, binding site, called the allosteric site. And this is where non-substrate molecules binds and induces the change of the enzyme shape. For an enzyme to function, it requires a specific substrate. This substrate will bind to a specific region on the enzyme. We call it as active site, as mentioned in the previous slide. So now, let's see how exactly substrate binds to an enzyme. To explain the enzyme-substrate interaction, there are two models or theories proposed. The first one is the lock and key model. So for this lock and key model, it is suggested that both enzyme and substrate have specific complementary geometry shape that fit exactly into one another, just like this lock and key. So if you are using a wrong key, you can never open the lock. So this model or theory was proposed in 1894 to explain the high specificity of enzyme. But sometime later in 1958, there's another theory proposed. They call it as induced fit model or theory. So this model suggests that the active site of the enzyme actually undergoes a slight alteration to accommodate the substrate. So now let's see the schematic diagram here. So there's substrate and enzyme with the active site. So from this figure, you can see that the active site of the enzyme is, the shape of the active site is slightly different from the substrate. So according to this model, a proper substrate is capable of inducing the proper alignment of this active site and allows the enzyme to perform its function. The binding of substrate to enzyme forms this enzyme-substrate complex. So this complex is temporary and the enzyme will always return to its original state after completion of the reaction. So now let's look at this figure on your right. So I have triglyceride and the 3D structure of a lipase. So this is the active site of the lipase, and triglyceride, the substrate will enter this active site and the lipase will catalyze or hydrolyze these triglyceride into fatty acids and glycerol. To see how substrate binds to enzyme using computer simulation, let's watch this video. The high specificity of enzymes towards its substrate, some enzymes require helper molecules to catalyze the reactions. These helper molecules are called cofactors. Cofactors include inorganic ions and non-protein organic molecule, or we call it as coenzyme. The word co means it works together with the enzyme. Inorganic ions such as magnesium, calcium, copper and iron, they assist directly in catalysis. For example, DNA polymerase and magnesium. DNA polymerase is an enzyme used to synthesize DNA sequence. DNA polymerase requires magnesium as cofactor. Without magnesium, this enzyme is unable to function optimally. Next, we have coenzymes. Examples of coenzymes are NADP+, FAD and NAD+, and vitamins. NAD+, NADP+, FAD are non-protein organic molecules that act as carrier molecules. These carrier molecules participate in the reaction and may even contribute atoms to the reaction. Coenzymes work by binding to the active site of the enzyme. Now let's see how cofactors and coenzymes work. Many cofactors will sit in the enzyme active site and assist the binding of the substrate. An inactive enzyme without the cofactor is called the apoenzyme, while the enzyme with the cofactor altogether is called holoenzyme. In the presence of cofactor, for example, we have calcium. The ion sits in the active sites and contributes to better binding of the substrate. Without cofactor, the enzyme will be almost certainly inactive. For coenzymes, since coenzymes and enzymes are non-metal organic molecules, they bind together by forming covalent bonds, means coenzymes share electron with the enzymes. When they form this bond, they only help the reactions to occur by carrying and transferring electrons through the reactions. When the covalent bonds are broken at the end of the reaction, the coenzymes return back to free circulation within the cell until it is used again. Just now, I showed you the schematic diagram on how cofactors and coenzymes assist in binding of the substrate. Now I want to show you this structure. So this is the crystal structure of DNA polymerase. So as I mentioned before, DNA polymerase requires magnesium as cofactor. So as you can see in the structure, there are two magnesium ions, colored in green. So one magnesium ion is called catalytic magnesium, means this magnesium assists directly in catalysis. And this study was published in 2011. Another example is NAD. So this is the malate dehydrogenase in complex with NAD. So this is one of the enzymes in Krebs cycle, and it is critical to cellular respiration in cells. In enzyme catalyzed reaction, the enzyme speeds up the rate of reaction by lowering down the activation energy. I believe you have learned this before, and activation energy is the amount of energy that must be added for a reaction to begin. For a reaction to occur, it is necessary for the reacting species, be it atoms or molecules, to come into contact or to come together or collide with one another. However, molecules frequently do not react with one another unless they are activated in some way. So, enzyme plays its part by bringing substrate into contact. Enzyme binds to a reactant and facilitates its transformation into a product. So, enzyme catalyzed reaction pathway has a smaller energy barrier to overcome before the reaction can proceed. You have seen how enzymes work. The enzyme activity can be affected by temperature, pH, substrate concentration, and many other factors. But for this lecture, I will explain on these three factors. So these are the typical graph showing the enzyme work best at certain temperature. For example, here at 37, at pH 7, and this one at certain concentration of substrate. Okay, now let's look at the details. First is on temperature. So increasing temperature increases the kinetic energy that molecules possess. So this causes more effective collisions between enzyme and substrate. But as temperature increases, more bonds, especially the weak bonds, will break. Breaking bonds within the enzyme will cause the active site and the whole structure to lose its conformations. Sometimes we say that the enzymes unfold. So eventually the enzymes loses its function and the rate of reaction will decrease. Okay, next is pH. So different enzymes have different optimal pH. So any change in pH above or below the optimal pH will quickly cause a decrease in the rate of reaction. The bonds within the enzyme structure can be influenced by hydrogen and hydroxide ions. So since these two ions are charged, so they may interfere with the bonds that hold together an enzyme. So this interference causes a change in the shape of the enzyme structure, including the active site. Okay, now the last one is substrate concentration. So same like temperature just now. Increasing substrate concentration increases the rate of reaction. This is because more substrate will be colliding with enzyme molecules, so more product will be formed. If you still remember the graph for substrate concentration, it looks like this. So after certain concentration of substrate, any increase will have no effect on the rate of reaction because the enzyme will be saturated and will be effectively working at their maximum possible rate. So the graph becomes plateau. Yeah, so this is just to share with you the current knowledge on enzymes. Maybe before this you learned that enzymes work best at 37 degrees. Yes, that is true for human enzymes and also for mesophile. But we do have organisms that live in extreme habitat. So these organisms, they produce enzymes that are active even at 100 degree, 0 degree, very acidic pH and very alkaline pH. The presence of enzymes and the absence of enzymes, as well as the concentration of enzymes, can be regulated by our cells. Cells can even activate or deactivate some enzymes. So this is very important because our cells produce or synthesize enzymes to catalyze metabolic reaction when necessary. Although enzymes are required for almost all metabolic reactions, enzymes need to be tightly regulated to ensure the levels of the product do not rise to undesired level. This is accomplished by enzyme inhibition. There are two types of inhibition: reversible inhibition and irreversible inhibition. For reversible inhibition, the word reversible means the inhibitor transiently binds to the enzyme, while irreversible inhibition, the inhibitor binds permanently to the enzyme. For example, we have styrene, warfarin, and cyanide. So these inhibitors binds and may inactivate and destroy the enzyme completely. There are two types of reversible inhibition: competitive inhibition and non-competitive inhibition. For competitive inhibitions, competitive means both substrate and inhibitor competes for the same active site. So the products form only when the substrate binds to the active site. So if inhibitor binds to the active site, the products are not produced. On the other hand, for non-competitive inhibition, the inhibitor does not bind to the active site, but it binds to the allosteric site. So if you still remember, allosteric site is the binding site on the enzyme structure apart from the active site. So binding of this inhibitor to the allosteric site causes or induces the change in the shape of the active site, so making the substrate unable to bind to the active site. On the other hand, for non-competitive inhibition, so the inhibitor does not bind to the active site, but instead it binds to the allosteric site. So if you still remember, allosteric site is the binding site other than the active site. So this the binding of inhibitor to the allosteric site causes or induces the change in the shape of the active site, so making the enzyme unable to bind to the substrate. This is the schematic diagram for reversible inhibition. So let's see this one. This is condition A, where there is no inhibitor. So when there is no inhibitor, substrate can freely bind to the active site of the enzyme. Next is B, condition B, when there is inhibitor. So when the inhibitor binds to the active site of the enzyme, substrate cannot bind to this active site. So this is called competitive inhibition, where both substrate and inhibitor competes for the same site. The last one is C, the condition where the inhibitor binds to the site other than this active site. So this is called the allosteric site. When the inhibitor binds to this allosteric site, it induces the change of the active site of the enzymes, hence the substrate is unable to bind to the active site. You can also find this figure from your book. So this is an example when the end product of a metabolic pathway becomes the inhibitor for the enzyme. So by inhibiting this enzyme, the substrate cannot bind to the active site, so the product production is stopped. This is the crystal structure of an enzyme with an inhibitor. I hope by showing you this structure, you can get a better understanding on how exactly enzyme inhibitor complex looks like. So we have here is the bacterial lipase complex with orlistat, the inhibitor. Because the inhibitor has almost the same or similar structure to the substrate of this lipase, the inhibitor can bind and fit in the active site. So when the inhibitor is bound to this active site, substrate can no longer enter the active site. We have finished with enzymes, and this is the last subtopic for this lecture, organelles and flow of energy. In the first part of this lecture, I've explained on the flow of energy in general, where we have plants absorb solar energy and converts it into carbohydrate and oxygen during photosynthesis. Photosynthesis involves this organelle chloroplast. So the products of photosynthesis will then be taken up by animal or as human to be broken down to generate ATP. And this conversion happens through a process called cellular respiration, and it takes place inside mitochondria. These are the equations for photosynthesis and cellular respiration. So from these equations, we can see clearly that oxidation and reduction reaction happen. Let's watch this video to recap the oxidation and reduction concept. So basically what you have to remember for oxidation reduction reaction is this oil rig. Oxidation is loss and reduction is gain. That's the end of this lecture. To summarize this topic, we started off with the flow of energy and food as source of energy, and followed by ATP as energy currency, where you learn about the function of ATP and also ATP cycle. And for the second part of this lecture, it is on metabolism and metabolic pathways. So you know that enzymes are involved in these metabolic reactions. So we get to know more about enzyme structure and the factors affecting the enzyme activity. And we end this lecture with the organelles involved in cellular respiration and photosynthesis, and a little bit on redox reaction. So thank you for listening. I hope you enjoyed this lecture. If you have any question, please do not hesitate to contact me or your lecturer. That is all. Thank you very much. Assalamualaikum and stay safe.